Hi, I'm Randy Reed, Executive Director of the National Lighting Bureau. Let's Talk About Light and Health is the name of our series, which is co-hosted by Mount Sinai's Light and Health Research Center. This segment is entitled, It's a Complicated World Out There, Exposomics and Light. And I am joined by Dr. Bob Wright. Dr. Wright, welcome.
Welcome, Randy.
Well, thank you for joining us today. And tell our audience a little bit about your background, please.
So I'm a pediatrician, an environmental epidemiologist. I went to medical school at University of Michigan. I trained in pediatrics at Northwestern and toxicology and environmental health at Harvard. And I came here to Mount Sinai about 10 years ago. I'm now the chair of the Department of Environmental Medicine and Public Health and the co-director of the Institute for Exposomic Research.
Okay, wow. Well, thank you very much for that and for telling us about your background. What is exposomics? What does it mean?
So exposomics is the study of all the health relevant environmental factors across our lifespan. So people are familiar with genomics. Genomics are really the study of all the genetic DNA sequence inside your body. The major difference between the two is your DNA sequence is the same the day you're born as the day you die. It never changes, whereas your environment is changing. So it changes on a daily, weekly, or annual basis, even seasonally. And it's for that reason a bit harder to measure than your DNA sequence, but at the same time, it's far more likely to be more important than your DNA sequence because your DNA never changed. It's the same as it was 20 years ago, 30 years ago. It's the same it's going to be tomorrow, next year, or 10 years from now.
DNA does not change. Exposomics does change.
Yes, over time. Yes.
Over time. And again, define it though. Define exposomics.
Well, there's things that we can measure directly in the body using chemical instruments that measure chemicals that are nutrients, chemicals that are things like pesticides or lead or mercury, things like that. And those are measured in laboratories. And then there are things that there is no laboratory test for, and light is one of them. Air pollution is another, sound is another. Stress really doesn't have a clear measurement that you can do in a laboratory. So exposomics has two areas. One we call the external exposome, and light would fall into the external exposome. So would sound and so would air pollution. And then there's the internal exposome, which are the chemicals that we can measure inside your body.
Okay. I'm starting to understand a little bit. Well, what got you interested in this to begin with?
Well, I trained in toxicology and I'm an environmental scientist. And the way that environmental health has been studied over the years has been to study sort of one thing at a time, whether it's stress, whether it's lead poisoning, whether it's mercury. We just study one thing. Well, there are millions of environmental factors that we experience in our lifetime, and they change from the time we're born to the time we die. The food that you eat when you're a child, when you're an infant, is very different than the food that you eat when you're a teenager, or the food that you eat when you're an adult. And though that food has different nutrients, it also has different types of pesticides and environmental exposures. The way you behave, babies crawl on their hands and knees and they put their fingers in their mouth. That changes what their environmental exposures are. Teenagers have risky behaviors. They may be more likely to drink alcohol or do other things that will change their internal exposome. And then adults have different diets when they're 20, 30, 50. They have different behaviors. They live in different parts of the country, different parts of the world. They live near sea level. They may live very high up. If your altitude is very high, the amount of oxygen you breathe is a little bit lower, and it changes the inside of your body and the chemicals that are in there. So all these things have effects on our life, and they're largely being unmeasured. So while genomics has had the idea of measuring all those genomic features, exposomics is really trying to do a similar parallel measurement of the exposome, of all the environmental factors that impact our health.
Which to me seems nearly impossible.
It's actually far more possible than you think. So first of all, I would say if genomics had the idea that measuring 4 billion base pairs, because that's how many base pairs are in your DNA, was impossible 30 years ago, and I think people probably would have said that, we wouldn't be where we are today. So I don't think measuring the exposome is impossible, and I'll tell you why. There's a number of things that are happening. So almost every person in the United States has a cell phone. So cell phones are GPS devices. So right now, Apple and Google know where you are because you have your cell phone with you, more likely. They can even tell whether you're indoors or outdoors based on the strength of the signal that the cell phone is receiving and transmitting. So if we know where you are, we know the quality of the air you breathe, because there are monitors everywhere that are measuring that. We know probably how much light, we can at least estimate it, how much light you're getting, because we know whether you're indoors or outdoors. We know whether it's a cloudy day. There are different measures that we could bring together that would let us estimate how much light you're being exposed to, how much sound you're being exposed to, how much air pollution you're being exposed to. And we can do that on a minute by minute basis in theory because of this cell phone that tells us exactly where you are every minute of the day. And right now we have environmental instruments that can also take a sample of blood or a sample of urine and measure tens of thousands of chemicals in your blood or urine. So we can know the types of food you ingested, the types of pesticides that may have been on those foods, the types of chemicals and products like perfumes that you may be using. All these things actually can be measured. What we haven't done is invested as a society in bringing all these different factors together so we can measure the exposome. I would argue that all of the technology needed to measure the exposome already exists. We just haven't invested to bring it all together.
Okay, what are some of the specific studies that you've been involved with?
So I'm part of a large NIH program called HERE, the Human Health Exposure Analysis Resource. So we're a consortium of laboratories that measure exposomic assays in blood or urine. So we measure thousands and thousands of chemicals in blood and urine, and we do it in different populations around the country. Exposomics is a relatively new field. So one of our challenges is just defining how the exposome varies in different scenarios. So if you live in a suburb, your exposome is different than somebody who lives in a city versus somebody who lives in a rural area. And as I mentioned earlier, if you live near sea level, your exposome will be different versus somebody who lives in the Rocky Mountains because of the differences in the geography. Geography plays a big part in the exposome, far more so than the genome does. It will actually vary quite a bit. And so one of the first things we're trying to do is establish what is the average exposome in different scenarios.
Okay, how does light play a role?
Well, light is clearly part of the exposome. Light plays a big role in what are called circadian rhythms. So these are the rhythms that your body has, or the reason why we sleep once a day or for roughly 8 hours are due to circadian rhythms. And those can be disrupted by light. And when they get disrupted, they have lots of downstream effects. Some of those downstream effects are actually on the metabolism of chemicals. It can also cause stress. It can also cause other types of health impacts that intertwine with the exposome. Also, keep in mind air pollution. We're very interested in air pollution and its effects on the body. One of its effects is actually to remove light. So air pollution makes light hazier. So different wavelengths might get reduced. So one of the ways that air pollution may affect the body is through its impact on light. So there's many ways that light intertwines with the exposome, as well as being part of the exposome.
So I read a study that said the effects of medicine are affected by the time of day because of the circadian stimulus. And that if you take a certain drug in the afternoon, it may react different or it may act different than you did in the morning. What about what you're talking about with geography? Could medicine have a different effect if I'm in the Rocky Mountains versus if I'm in a lower altitude area?
Oh, absolutely. Like for one example, we know that the risk of having a stroke is actually slightly higher. It's not a huge increase, but it is slightly higher if you live at high altitude. And so the reason is when you live at high altitude, there's a little bit lower percentage of oxygen in the air. And so that lower amount of oxygen in the air makes your body create more red blood cells, the cells that carry oxygen. So because you have more red blood cells in your body, it's slightly more viscous, a little more sludging can occur. And that sludging over time can increase your risk for having a stroke. So blood thinners will work a little bit less well in a place that has high altitude than a place that doesn't. Now, it's not a very huge amount that you can see on a one-to-one person basis, but if you took a group of 1,000 people on blood thinners in Colorado versus a group of 1,000 people on blood thinners in New York, which is right at sea level, you would find that on average, it works slightly worse in Colorado because of the increased amount of red blood cells in the body because of the altitude.
Okay, so we're mid 2022. Where will we be with Exposomics in 2027, in 5 years?
So I think Exposomics is at an inflection point. If you think about the advances in medicine since the Human Genome Project was finished 30 years ago, they're actually pretty small. We don't have a better idea of what causes autism. We don't have a way to prevent asthma through genomics. We don't have a way to stop Alzheimer's disease through genomics. The missing piece of the puzzle is really exposomics. And I think there's a growing realization that if we don't measure something beyond the genome, we're never going to fix all these problems that we have because it's not getting better. So, and I think NIH and I think a lot of researchers starting to realize that exposomics is the missing piece of the puzzle. I would say in terms of where we are, we're about 15 years behind genomics. So we're roughly where genomics was 15 years ago. 15 years ago, we could not sequence, a person for less than $1 billion because that's roughly what it costs to do the first 6 people. Now we can do it for less than $1,000. So I think we're going to be able to measure our exposome in about 15 years for less than $1,000. And I think it's going to be through a combination of the external exposome. So creating maps that show where air pollution is and how it varies day by day and linking it to where you are using a GPS device like your cell phone. There'll be ways for people to get information on their exposome that way. And I think there's going to be ways where people can go to the doctor, they can give a blood and urine sample and have an output of the tens of thousands of chemicals that are inside their body and whether or not it's changing their risk for a disease.
So, the main goal of this is light and human health and to help people. But I almost see a commercial aspect of it. I can see Exposomics Tourism. Go to an exposomic friendly area. Do you see things like that happening in maybe 20 years?
Well, I'm a physician. I'm not an entrepreneur, but I do think that the private sector will play an important role because I think one of the ways in which genomics grew very quickly was because of companies that wanted to leverage genomics in their businesses. And I think that's going to happen with exposomics. I think cell phone apps are going to play a big role in that. But I also think there's going to be, as you said, people who want to pay money just to get their exposome measured. And I think there'll be ways to do that.
Dr. Wright, what can people do right now to improve their exposomics?
Well, some of the things we talked about, like cell phone apps, already exist. So there are apps that will tell you about the quality of the air that you're breathing. There are apps that you can actually use to try to find information about what chemicals are inside a particular commercial product and whether or not they have any known health effects. So those things already exist. I think they need to be expanded and people need to become more aware of them. It's not widely known that they exist, but there are things that people can do. If you can afford it, I would recommend eating organic. Organic foods will have less pesticides, and I think that would be good for you. I hesitate to advocate strictly for organic because I want people to eat fruits and vegetables, even if they're not organic, because it's healthier than the alternative. When you can use these apps to decide whether or not it's a good day to go exercise. You say you have asthma, but you still want to jog. There'll be good days and bad days with respect to air pollution, and the apps can tell you what are good days and bad days. So there are ways that you can employ this information in your day-to-day life.
Well, Dr. Wright, this is fascinating. Thank you. And please hold on for our questions. We're going to now open the floor for questions. And please do use the Q&A button to ask your questions.
welcome to the live discussion of the Let's Talk About Light and Health series. I'm Alison Thayer, a researcher at the Light and Health Research Center. I'm joined today by Dr. Robert Wright. Thank you, Dr. Wright, for that really nice interview. To start off, I wanted to ask about one of the quotes that you had said. You said, "The technology exists, but needs to be expanded and more well known." So how do you see all of these apps coming together? So things that track your sleep, your nutrition, the air quality, and even tracking the light that you have in your space. How do you see all these coming together?
Well, I could answer that also with respect to light. One of the difficulties I imagine, with light and health is, outdoor light is very different than indoor light, and indoor light requires being measured at an individual level. But, I don't know that we necessarily have to measure it in every person. If we do studies where we've measured indoor light in people and we know different characteristics about those people, their age, other demographic factors, their occupation and such, or even have measurements in their occupation, we can start to make maps of what the indoor light is like in different areas. So we can start to make maps of what indoor light is like inside apartment buildings in New York City versus Seattle, inside homes in the suburbs versus inside rural homes. And as we start to make these maps and we start to plot the distribution, we'll probably have a better understanding of how indoor light gets distributed across people. And we may even be able to use statistics to sort of impute what the average indoor light is for a person of a given age and a given demographic living in, say, a Seattle suburb versus the Seattle city versus, say, a rural area in the state of Washington. I imagine that there's differences in all those areas, and we don't necessarily have to measure all millions of the millions of people that represents. We can measure a few thousand of them, and we can probably use statistics and AI, artificial intelligence, to actually begin to map what the average person is like, and their light exposures like in those different areas. And I think that's probably one of the ways it's going to come together.
That's great. And yeah, a lot of the stuff that we do to measure light specifically, it changes so much depending on the space. An office space is really different from a patient room. And even within the same room, if you move closer to a window versus more in the room, or different locations you are in respect to a light, it really changes what exposure you're getting. So I feel like it kind of has to be on a really individualized basis as well.
Yeah, although I would bet that there's probably a pattern, a covariance pattern, to use a statistical term, of how lights in given settings tend to covary. So lights in the average doctor's office will probably be more similar than, say, lights in an accountant's office. And there may be ways to mathematically sort of impute what the average light exposure is, so that you don't have to measure all 350 million people in the United States. You can measure a subset of them and make estimates, very educated estimates of what everybody else is facing or experiencing, and then you can relate those to health impacts across a larger population. So I do think there's a role for actually using artificial intelligence to estimate exposures, but you're going to have to collect really good data on a subset of people in order to estimate what that exposure is on the other people. And actually, that's exactly what genomics does. People don't realize this. Very few genomic studies actually sequence the entirety of their DNA. They measure a subset of a person's DNA and they impute mathematically what the rest of it is. And so we can just borrow from those methods as well to sort of estimate what the average light exposure is in different populations.
So can we talk about the different factors of exposomics? So, what do you find are the most important? We've talked about, a bit about light pollution, pesticide exposure, air pollution, altitude, even circadian entrainment. Can you, do you have type of rank order for these, or do they all play a role together?
Oh, well, nothing happens in isolation. So all these things tend to come together. And it's sort of like what I would envision is what can sometimes be called an exposomic risk score. So in genetics, they have these polygenic risk scores where they measure your genome and they count all the genetic variation in your DNA that might be associated with the disease, and they kind of add it up. I think that's probably where exposomics is going. So we're going to try to measure all these different health factors that we can measure in the exposome at the individual level for a person, and then add them up to create a risk score. So I think that's how it comes together. But in terms to answer the rank order, to be honest, I think the most pressing problem facing the human race is climate change. So I would probably rank that number one because it's going to affect everybody on the planet. And it's going to have impacts that we don't understand that are downstream. We're going to see if the climate warms in temperate areas like the northern United States, we're going to see different species of mosquitoes there, and different species of mosquitoes may carry different diseases. So we may be seeing changes in diseases that are actually due to climate change. So there's going to be all kinds of things that are going to be hard to predict. Or if there are more mosquitoes, people are going to use mosquito repellents more, and there may be a health cost to that as well. So there's all kinds of things that I think are very difficult to predict. What sometimes people call butterfly effect, the flapping of a butterfly in theory and chaos theory potentially causing a hurricane. There's all going to be all kinds of downstream effects that we probably won't be able to predict, but will probably, once they occur, start to put the pieces together as to how they occurred.
We're also asked, in your view, are there any environmental exposures comparable to smoking? So what other environmental factors should we be cautious about?
I think air pollution has been underestimated in terms of the toll that it takes on people. There's a lot of literature now that's starting to look at the role of air pollution in diseases other than lung diseases. We used to think that it was primarily just affecting our lungs. And then we realized it's also, playing a role in heart attacks. Now it looks like it's playing a role in Alzheimer's disease and other kinds of diseases that elderly people get, Parkinson's disease. It plays a role in child development, particularly brain development. There's even some evidence that because air pollution causes chronic inflammation, it may actually predispose people to obesity. So there's all kinds of issues that air pollution plays a role in that are very, very important. And perhaps what we don't appreciate is, we're all breathing similar air to the other people around us, is that it's having an effect on large numbers of people. So even if it's a relatively small effect compared to smoking, which is a very big effect, it's affecting almost everybody on the planet, air pollution, similar to climate change. And so as a whole, it's actually causing a lot of problems at the population level. So I would probably rank air pollution up there.
So there's been a lot of talk about light at night and increased cancer risk. Is it hard to disassociate light at night from other behaviors? And how do you do that with exposomics?
Well, in order to parse out the effect of light at night, I think you need to measure the other environmental exposures that are correlates of it. So I think exposomics will help actually answer that question. I don't know the answer, but I think by measuring the other factors that tend to correlate with light at night, temperature variation could be one of them. Obviously, there's stresses on the body from being, if you work shifts overnight. All those things need to be measured because they will vary from person to person and what role they play, whether or not, I'm not even sure, to take a step back, that they're necessarily alternative explanations. I tend to think of it as more like a complex mixture of different environmental factors. Light at night tends to co-occur with stressors because you're up all night. A lot of jobs, not all jobs, but some jobs where you're up all night, such as nursing, actually are quite stressful. So you have a combination of light at night and stressful, social situations or occupational situations, and the combination of those may actually be more toxic than just simply being awake all night. If you're a night watchman and not much happens, and it's actually kind of a dull job, that might not be the same as an ICU nurse who's up all night dealing with stressful situations. So I think the combination of these exposures really is something that we need to start looking at.
So going back a little to there's a follow-up to the question about the phone apps. So with so many different variables being tracked from person to person, how do you control for the confounding factors in such large data sets? And which variables have you found are the most heavily influenced in subjects?
Oh, that's a great question. We always worry about confounding in epidemiology, although I think a lot of times what we think of as confounding really isn't confounding. It's more of a combination effect, which is similar to the way that genomics looks at multiple variants. Instead of thinking of them as sort of being alternative explanations, perhaps it's the cumulative impact of all of them. So similar to my previous answer about light at night and stressful situations, I think a lot of what we measure in the environment aren't really alternative explanations. They're part of this sort of complex mixture of things that happen to us that all have effects on us, both good and bad. You know, one of the goals of Exposomics isn't just to find out the scary things that we're potentially exposed to, but are not aware of, like pesticides on foods, but also the positive things. You know, there's a lot that can be said about the social environment and social networks that actually promote our health. There's a lot about nutrition that actually promote our health. So we equally want to understand what those things are so that we can give people advice on not just what to avoid, but also on what to do.
Can you relate the endocrine system, since it's linked directly to light in our bodies, specifically to these measurements?
Sure. Yeah, so the assays that we run, one set of them are called untargeted assays, and these actually capture or try to capture all the chemicals inside the blood sample or the urine sample that we measure. So they would include all the measures. So cortisol, thyroid would be picked up, sex hormones would be picked up, dopamine, basically all of them. And so we can look at their various levels as they relate to each other. So there may be some advantages to that. You know, cortisol has a diurnal pattern, as you guys know. So you know, measuring it at one time point in the day is not as valuable as measuring it serially over the course of the day. But perhaps one way that exposomics can add to this, at least with assays, is that we can look at the cortisol in relationship to the other hormone systems. And hormones don't really act in you know, isolation, so they do coordinate a lot of their excretions. So we can actually look at cortisol as it relates to many other hormones simultaneously. So there may be some added value to an exposomic approach to hormones in the endocrine system.
We have a couple questions on sunlight and specifically ultraviolet light as well. So kind of the two questions together I'll ask, but how important is exposure to ultraviolet light? Are there pros and cons? As well as can exposure to sunlight reduce the effects of exposomes or exposomes accumulative?
Well, yeah, exposure to light is very important for many health effects. There are people more expert than me on this issue on the call, but you know, certainly vitamin D is an example. Exposure to sunlight is very important for vitamin D. Vitamin D plays a big role in many different systems inside our body, so it can actually play a role in things as varied as bone health and asthma and lung health. So sunlight is very important. In terms of wavelengths, there are different wavelengths that matter for different systems and outcomes, including sleep. So measuring light is very complicated, just as measuring the exposome is very complicated, and all these factors have to be taken in.
There's a difference too between, you know, we talk about a lot of how light gets through the eye, but you know, with sunlight too being outside, you get exposed on your skin and kind of get a little bit more of that UV ultraviolet there. So you know, there's those two different factors as well.
And there's the potential toxicity. Obviously, sunburns or skin cancer are a downside of UV light. So, you know, they're like most things, if not all things in the exposome, you know, it's not just whether or not you get exposed to something, the dose matters too. So overexposure or over consumption of anything can be toxic, as well as underexposure of nutrients. And I think light kind of fits like a nutrient in that, and that too little light or too much light can both be toxic. You really want to try to titrate the correct amount.
Just a couple more questions. Do you think that exposomics will be helpful at determining at what time of day we can increase light levels to get positive circadian entrainment in buildings? It's kind of more of an application question.
I think exposomics can play a role. Again, I think there are folks on this call. Dr. Ferreira is an expert, and I think could probably better answer that question than me. But I think the role that exposomics can play in tandem with light measurement is doing things like measuring, different metabolic responses to light. There's going to be changes not just in our endocrine system. There's going to be changes in our metabolism. And these are all part of the exposome. And I think some of the measurements that we make, particularly in assays, can be very helpful in better understanding the role of light, and maybe even figuring out if there's a particular time of day in which particular wavelengths are most valuable. And I think those sorts of measurements where the exposome can actually be very, very helpful for, because the exposome isn't just the external world, it's also the body's reaction to that external world in terms of its metabolism and its endocrine response.
So we're getting a lot of nice feedback just on your presentation, how nice it is. And one last question too, just to kind of wrap up. Some participants want to learn more. So is there a site that they can go to learn, or have different materials that you have?
If you Google Mount Sinai Institute for Exposomic Research, you'll find our site. I actually write a monthly blog. I think it's in my email signature now, but you can subscribe to my blog. So that would make me very happy if I got more subscribers after this call. So yeah, there is a lot that can be learned from both those websites. And also, as I said, exposomics is becoming of greater interest nationally and internationally. So there's other resources as well if you were to Google exposomics.
Great. Well, thank you so much for your time, Dr. Wright. And thank you all as well for those great questions. It was a really nice discussion. And we look forward to seeing you next month.
Great.
Thank you, everyone.
Thank you all.